this group, the basic and translational cancer research. The group is chaired by Dr. Jack and Rotimi, and it aims to understand African cancer fundamental biology and generates and test new ideas, principles, and theory about the disease that affect indigenous African. The group intends to bring all African scientists together to know each other and collaborate to address pressing questions, needs, and resources concerning cancer research in Africa. The goal and the strategy of the group is to create a platform for African researchers to network and collaborate to share expertise and find solutions, integrate institutional, national, and regional level data to begin to understand the biology of African cancer on a continental scale. Form collaboration among researchers and similar interests to apply for grants to enhance their research, train researchers on grantship, publication writing, research ethics, and etc. So with this brief introduction, it is my pleasure to introduce our first speaker for this seminar, and hopefully we'll be having more seminars. And I would love if everybody who's attending to send their email and their interest and to tell us what they are doing, what type of research they're doing, and what is their goal for the future, so we can form this cohesive group. So the first speaker is Dr. Evelyn. She is assistant professor at Henry Ford Health System. She's originally from Ghana, and she will talk about opportunities and challenges for basic cancer research in Africa. And Evelyn, the floor is yours.
Oh, sorry, can you hear me now? I guess
yes.
Okay. Thank you so much, and thank you everyone for making time to join us today for our first webinar. My talk is more of an introduction to why it's important to do basic cancer research on a continental scale in Africa. And so I'll use the next 15 minutes to give a brief introduction on the work we are doing and why it's important for all of us to come together to understand the biology of African cancers. So the title of my talk this morning is Opportunities and Challenges for Basic Cancer Research in Africa. Okay, and my presentation will have this outline. I'll talk about the need for basic cancer research in Africa, understanding the biology of TNBC, or triple negative breast cancer, in women with Western Sub-Saharan African ancestry, which is the work that we do in precision medicine for aggressive breast cancers. We talk about opportunities to build basic science research capacity in Africa, the challenges we have, the current state of basic cancer research in Africa, and the role of this special interest group. Why is there a need for basic cancer research in Africa? Is it a necessity or is it a luxury? Basic cancer research in Africa, for the purpose of this presentation, will refer to basic and translational research into continental African cancers that is occurring in Africa and being conducted by Africans. And so when I refer for this, the purpose of this presentation, it doesn't refer to any form of research that is being done on African tumors by any researcher outside Africa. A patient's ancestry is known to contribute to the frequency of the genomic alterations that occur in that patient. And so the kind of cancers the person develops, and it affects the prognosis and the progression of the tumor. For example, patients of African descent with breast cancer have earlier onset of disease. The disease tends to be more aggressive and a higher mortality rate compared to other ancestries. Still talking about the need for basic cancer research, we need to build a foundation of African-specific knowledge. that is understanding the biology of all cancers that happens in Africans for the purpose of translation into the clinic and also to influence policy. This will also help in allocation of resources, both within the African continent and also outside Africa. For building a foundation of African specific knowledge, we need to understand the biology of the tumors that happens in Africans. African cancers have been known or have been reported to be biologically unique. That is, the biology and the mechanisms that control the cancers that occurs in Africa, at least for prostate cancer, for breast cancer, for other cancers that some of that have been published on, we know that the biology is different from what happens in other ancestries or in other parts of the world. And so it doesn't benefit Africans when we extrapolate findings from other populations and other ethnicities to Africans, because we don't conduct basic cancer research. We need to understand the mechanisms of the cancers that happens in Africa for translation and also for implementation. I will use an example of what we have studied in triple-negative breast cancer to make this point, that we for the purpose of understanding the biology of triple-negative breast cancer in women with Western Sub-Saharan African ancestry. Our team, which is Precision Medicine for Aggressive breast cancers, use the approach of bringing researchers together from countries both within Africa to partner with other people outside Africa for the purposes of gaining resources and funding to study African breast cancer in Africa. We have a collaboration of 10 institutions in Africa, both in Western and East Africa, and hoping to have other people from other parts of Africa to join us in the work that we do. So for the purposes of understanding the biology of triple-negative breast cancer, which is breast cancer that is most prevalent among women with Western Sub-Saharan African ancestry, and it is the most aggressive form of breast cancer. For the purpose of this presentation, it does not express the estrogen receptor, the progesterone, or the HER2. It does not overexpress HER2, and so it is very difficult to treat it, and chemotherapy is the mainstay of treatment. And because it's most prevalent among Africans or Western Sub-Saharan Africans, we need to find why it is most prevalent among this population and how we can treat it. What is the mechanism? We have reported many times that it's prevalent amongst us, but why is it prevalent? And what controls the biology of the tumor, and how can we treat it? So for the work we do, these institutions come together. We have IRB approvals. We operate on the patients, and with consent, we take tumors from these patients. The tumors, each institution that participates in this work has the resources or have been given the capacity to have a cold chain system within the institution. They have their minus 80 freezer, liquid nitrogen, everything available on site. And so the tumors are stored both in formalin and also snap frozen. We are able to extract DNA and RNA from these tumors for sequencing. And then we are able to also implant some of the tumors in mice to generate patient-derived xenografts, and also develop cell lines from some of these tumors from women with African ancestry. So this is work that is being done on African tumors in Africa. But then we partner with institutions now in the US because of some of lack of facilities that we need for some of the work that is needed. And so from the initial work that we have published, just because of time, we will not go into all the details. We are beginning to see that tumors in women with Sub-Saharan African ancestry have a distinct immune signature that regulates these triple-negative breast cancers that can be that can be targeted for therapy. When you compare Ghanaian to Caucasian tumors, we have some of these pathways that I'm showing, the ECM receptor interaction, nitrogen metabolism, antigen processing presentation, PI3K, AKT signal pathways coming up in the Ghanaian tumors. When you compare even with the African American tumors, which historically are used for studies that are extrapolated to African continent, we are realizing that when you take continental African samples, the signature in those tumors is very very different from African American tumors. And so if you extrapolate findings from African American tumors to Africans, it is not going to benefit us. And we need to study the tumors that occurs in Africa. We look at other, when you go further to look at the genes, the specific genes that are expressed in the African tumor, in this case, the Ghanaian tumors, compared to both the African American and the Caucasian tumors, we see that the genes that are being overexpressed are very different in the Ghanaian tumors. Again, buttressing the importance of us studying these tumors as they are, and not taking findings from other places. Now we are beginning to look at very specific genes. CD274 is PDL1, which is very important immune regulator. And now we are beginning to study that in our team too, because we are realizing that when we went further to look at the pathways that regulate PDL1 expression in Ghanaian tumor, we saw that toll-like receptor 9 expression through NF-kappa B pathways upregulate PDL1, specifically in Ghanaian triple-negative breast cancer. And this is very interesting because toll-like receptor 9 is a gene which is upregulated by malaria pigment. And considering that malaria is endemic in Ghana, we want to understand why toll-like receptor 9 is regulating PDL1 expression to the NF-kappa B, which means that it's even possible that the infections that we have in Africa are regulating the tumors that are occurring in Africans. And this is what we are currently studying, how the toll-like receptor 9 regulates the immune pathway through NF-kappa B. And we only saw this because we specifically studied the Ghanaian tumors. In another study that we had done, which we just submitted to Genome Medicine, here we looked at 333 tumors across all cancers to look at the genes, the differences in the gene expression in different tumors. And again, for the sake of time, I'll not be able to go through the findings, but it's very interesting to see that across all cancer types, each one, this is for breast cancer, we realize that the genes that are expressed in the Africans, that is in the yellow, is very different from the ones that are expressed in African American, East Asian, Europeans, or West Africans. And so again, buttressing not just in breast cancer, this is for breast cancer, and again, this is for prostate cancer. The genes that are expressed, if after the presentation, depending on the work you are doing, if you are interested in any of these genes or markers, I'll be happy to share it with you if it is relevant to your work. For the sake of time, I'll not be able to go through in non-small cell lung cancer and pan-heme and all other cancers. We are seeing that the gene expression is very different in Africans, in West Africans, in East African, in Southern African tumors compared to other ancestries. And so there is the opportunity for us to build the capacity for doing basic cancer research in Africa, because the African tumors have not been studied as much as other tumors have been studied. And so there's the opportunity to make discovery. I'm very mindful of my time. We have to build, we have the opportunity for building research careers. There are so many people, young people in Africa who are enthusiastic about studying African tumors. And so this basic and translational group is yearning to bring people together to find the resources and the funding that is needed for people to build a research career in Africa, and so that they will remain on the continent and study African tumors. There is also the opportunity to form meaningful collaborations that are beneficial to African researchers. And I'll just say that by this, any collaboration that we are hoping to promote between African researchers and other people from other parts of the world is supposed to be one that is offering training opportunities for the African researcher, building infrastructure for research in Africa, keep African tumors in Africa. So we don't want to promote so much of research that is just transporting tissues out of Africa, and also offer grant opportunities for funds that will remain in Africa. There are so many challenges. Cancer research in Africa has largely been led by foreigners because of funding, resources, and the lack of expertise. But and also the African researchers who wish to return to Africa, they have had the training outside Africa, but there is no support at home. There are no facilities that can support the kind of research they do. The infrastructure and the equipment and the funding is not available. And that is one of the things that we are also hoping to address. The rules are also for funding for applies differently to funds that are sent to Africa. And that's something that I have recently come to realize, that there are so many funding agencies. I had an agency that I got a grant from for my institution that's Henry Ford, and also my institution in Africa. And the rules were very different. So if you get a grant in Africa, the things they require from you, the amount of money they give you, how they distribute the funding, is so different, and it's almost impossible to do the work. And I don't know why it applies differently, but I guess they might have their reasons, but these are some of the things that we are hoping to address so that funding will be made available for research in Africa. So the basic and translational research group, we are hoping to help with so many other things, and that will help with the work that is done in Africa. As Salma had said at the beginning, we want to entreat all of you to help us have a baseline understanding of the current state of research in Africa. When I study the literature, there isn't much that is reported on basic cancer research across Africa. So there is the need for an assessment of the resources that are available across the continent. And so for that reason, we are hoping to administer a questionnaire to everybody here, and also other people who might not have been here, for you to fill in, to give us what facilities you have, what human resources you have in your institution, in your country, the equipment, the funding, the institutional support, and other resources, so that we can have a baseline understanding of what is happening across the continent, how we can bring people who are doing equal things together or the similar things together to collaborate to do the work. And then we are also hoping for the basic and the translational research group. The aim, I mean, again, is here for the sake of time. I don't need to go over, but we want to create a platform from Africa researchers to network and to collaborate in order to share expertise and find solutions together. We want to integrate institutional, national, and regional level data to begin to understand the biology of African cancers on a continental scale, as Salma has said, and also to form collaborations among to train researchers on basic. Oh, sorry, something in the background. To train researchers on the basics of research, such as publication writing and research. And so for the sake of time, I rushed through my slides, but I'll be happy to answer questions at the end of it. And I want to thank all my team members and all the PIs and the coordinators and everyone who works with us. Thank you. I kept my time.
Yeah, Salma, am I good to go? Hello, Salma.
Salma, go, yeah.
Yes, am I good to go?
Yeah, awesome. Yeah, you can go, yeah.
Oh, okay. Yeah, thank you very much, Salma, and the organizers, and Evelyn. What a great job, opening up this webinar today. And I think you just made my assignment very easy, in that I'll be focusing my talk mainly on prostate cancer in Africa. And if those of you that listen to our with rapt attention, if it's possible for you to just pull out breast in all the talks you gave, and you slot in prostate, what I'm going to talk about is going to be very much similar. So And so if you look at, prostate cancer, what you see, just as she said, is that there's a lot of, just as you have for triple negative breast cancer, there's a lot of ancestry signature. And I won't spend so much time on this. It's a picture that we're all familiar with, that the burden of prostate cancer in African America sort of reflect what you have in the sub-Saharan African, subcontinent. But the question then becomes, what exactly is driving this, kind of, outcome, disease burden that we're seeing? What is clear is that from several studies, we know that, genetics and genomics play a critical role, in the risk and outcome of prostate cancer. And several studies and more studies are clearly identifying genetic ancestry, particularly those that relate to the West African coast of Atlantic Ocean, to be clearly responsible for increasing risk and the outcome of this disease. And so if we look at the body that you have, and they clearly show that if you match age for age, you clearly see that there's differences in the tumor biology if you compare white and black. And this has also been replicated in Nigeria, where it clearly shows that very high percentage of patients, about 30% within 2 years, develop castration-resistant prostate cancer. And one of the things that I've been talked about is that, perhaps the tumor is just behaves like that in Black people. So this report was published by a group from, I think, University of Lagos here in Nigeria. And they look at autopsy, prevalence of prostate cancer at autopsy in Nigeria. And what you really see is that the percentage is actually low compared to what is observed in other races, even when compared with, Caucasian and even African American. And so the conclusion from their study is that, well, it's not that the disease is always there and not detected early, but the issue is therefore that once the disease is detectable, it's so aggressive, and the outcome, it presents a very unique aggression. And so if we take ourselves back to the base, okay, I think I dropped off here. Sorry about my Wi-Fi. Yeah, so that takes us back to the work by Charles Oge that clearly showed that androgen is a major driver for prostate cancer. And what has been shown by a study published by Professor Dedino, who is the AORTIC Research Committee, is that you see the difference between the duration in testosterone between white and Black. And so if you look at the studies that have been done across Africa, and several studies have looked at androgen, this hypothesis, and looking at the variation in the genes associated with its metabolism. And what we see is that clearly these genes, of course, they were from here, but they are not really risk marker for folks, for men in Africa, but rather they are surrogate indicator of ancestry. And so if you then look at GWAS studies that have been done on prostate cancer in Africa, we're talking about a disease that affects a lot of, that the risk factor is being from Africa. And just as, Evelyn talked about it, you really don't have a lot of basic science work going on. So you can only identify 3 studies that have GWAS studies that have ever been done on prostate cancer in Africa. And so if you look at one in Tunisia, one in Ghana, and one in Uganda, the interesting thing is that if you look at the risk chromosome, you could see that the Tunisia risk chromosomes are completely different from those that you have for sub-Saharan Africa. And if you compare, the West African, Ghana with, East Africa, Uganda, the only commonality that you see is on chromosome 6. And the interesting thing with chromosome 6 is that chromosome 6, with the cytoband that was identified, has to do with something, around the neighborhood of HLA, which is a gene that is involved in immune response. Again, it takes us back to what Evelyn talked about, immune response. So, just to also show you, the amount or the deficiency, the depth of the work that we've done on prostate cancer in Africa. So that is GWAS that I showed. If you look at, exome sequencing work, just one exome sequencing work has been published from sub-Saharan Africa. And that was a study from South Africa. I believe they only sequenced 1, 2, 3, 4, 5, 6 individuals. So they did an whole exome sequence for 6 prostate cancer patient, prostate cancer tumor. Very interesting study, landmark, very interesting. And so recently, we just got this paper published in our consortium, and just to let you know that I'm part of the Prostate Cancer Transatlantic Consortium. We've sequenced about, I think we're able to sequence up to 50. The interesting thing you see there is that we have a very high mutation of BRCA2 gene, and when you compare the mutation frequency with African American with European, you clearly see that the Nigerian patient population that we study have very high mutation in BRCA2, APC, 8M, BRCA1, MED2, 12, and TP53. The interesting thing about this group of genes is that they are part of the mechanism that we can take advantage of in perfectly treating this disease. That there are the genes related to mismatch repair and homologous recombination. And we know that there's a class of drug that that the patient with this type of mutation responds to very well, and that is PARP inhibitor. The interesting thing is that in 2020, the clinical trial for olaparib for metastatic castration-resistant prostate cancer was published. And it showed that patients with castration-resistant prostate cancer were treated with PARP inhibitor or any brand, in this case, olaparib, responded very well than the enzalutamide abiraterone. The interesting thing, however, is that if you look at the patient population, they used 200 participants included in that study, and out of the 200 participants, was only 3, that were Black ancestry. And that is again throwing challenge, to us, Africa, to say, why can't we replicate this type of study in Africa and see how our patients, respond very well? And so, of course, based on that, FDA approved, olaparib for castration-resistant prostate cancer. So another consequence of, this type of, mutation in tumor is that it's also linked, to inflammation. In our study, and the foundation for the role of inflammation in driving prostate cancer, this racial disparity in prostate cancer, was actually laid by the work done by Dr. René Rims, and that was then built on by Stephen Arms, that clearly showed that African Americans who take aspirin very often have reduced prostate cancer aggression. And so building on that, this study by Adina et al. Went ahead to show that if you administer vitamin D to Black patients when they present for diagnosis, and the first biopsy was taken, after that they placed them on vitamin D for 2 months. What they found out was that after 2 months, the inflammatory signature of the tumor was completely removed. And that's, so we have the initial study by René et al. Then we have the study by Stephen Arms, which showed aspirin. And here we also have another study. And there are several other studies that have shown that if you're able to control for inflammation, you should be able to really address the aggression that you see in Black prostate cancer patients. And then, so working with Stephen Arms, collaborating with Clayton Yates, and I have one of my graduate students who work with them on this study. But then we're studying how vitamin D then plays a role in prostate cancer, particularly those of West African ancestry, West African men. And we clearly showed that, and this study is now submitted for publication, and you could look at it in bioRxiv if you are so interested. We showed that what vitamin D does, among other things, that regulates circadian rhythm, and also thereby also influencing inflammation, and also having some epigenetic regulation. And this epigenetic regulation is through the interaction, not just with vitamin D receptor, but with SMARCA1 and BAS1, and those are very important regulators. And we use immunohistochemistry to show this is, down-regulated, there's no expression of this in Nigerian prostate tumor. And then we look at the level of vitamin D, circulating vitamin D, the plasma of prostate cancer patients. Of course, we included colorectal patients in that study, and we clearly showed that the level of vitamin D, the median level of vitamin D is lower in prostate cancer Nigerian patients than you have. And so finally, as I begin to round up, I just thought I should also show this study where we completed the RNA-seq analysis of Nigerian prostate cancer patients. And when you compare with, and this is glycine-9, very high-grade disease, you compare with glycine-9 in TCGA of European America, it's completely opposite. You look at the heat map, completely opposite. The genes that you find, high regulated in Africa, in Caucasian, is completely down-regulated in Nigerian patients. and then you do the ingenuity canonical pathway, you could see similar pathway, to what, Evelyn talked about, the triple negative, breast cancer coming to play some role in driving aggression in high-grade prostate cancer. And we showed that this has to do with, natural killer cell, dysregulation. And so as I begin to round up, based on our study on vitamin D, we are now starting a clinical trial, which is supported by Mayo Clinic, to show how administration of vitamin D modulates the immune response of prostate cancer patients in Nigeria. And also based on the findings from the RNA study, we just secured funding of $10 million from DOD. And we're on a project, consortium project that we call IKEA. And we now continue, that gives us the opportunity to study how the dysregulation of the immune system, affects, poor quality of life and also alter the mental health of prostate cancer patient and survivor. I just briefly want to acknowledge our founders, and we'll be happy to take your questions. Thank you very much.
Thank you, Solomon. I'm sorry, I didn't introduce you. My computer is stuck. I can hear you, but I can't talk. So every time I hear you, I'm very impressed with your talk and your presentation. And so for the sake of time, please, if you have any questions, post it on the chat and we will take your question at the end. And now we can move to Dr. Montasser and then we'll take questions later. Dr. Montasser, the floor is for you. Hello, do you hear me? Hello?
Yes, we're gonna hear you.
I'm not sure, maybe Dr. Montassi has a challenge.
Yes.
Okay. Okay.
Great, good. Because I had just similar problem. My computer just stuck.
Yes. We hear you. Please go ahead.
Okay. Let me spend a couple of minutes on this slide because it summarizes a lot regarding the conundrum that we have now on are we going for personalized medicine? Is the age of the single gene, single protein phenotype is over? So this figure, which is taken from recent publications that came out in Frontiers in Genetics last month. It's a special topic. You have the normal curve. In the normal curve, on the left side, you see where the genetic effect is higher. And where you have the syndromes, the single gene higher the higher effect. On the right side, you have the effect of the environment and the lower effect of the genes. And here we have people who live a very reckless kind of life, but yet they don't develop any disease. But however, these are the extremities. In the bulk of what people encounter in the health and the medical service is in the center, and here where the gene environment interaction is at its highest. Here you have the interaction of all kinds of environmental hazards. You have the carcinogens, you have the viruses, you have the microbiome, which is also the microbiome is a state of flux. You know, it's a continuous kind of interaction. And we have seen now in the COVID how how these microbes alter our genome, because the genome is not about the gene, the genome is about the expression. So if you have a pathogen, if you have a bacteria in your gut that alters the gene expressions profoundly, then it is altering your basic genomic profile.
Muntasir, it looks like what you are talking about is different from what you are showing. I don't know if I'm the only one not seeing it.
So no, you don't see the slide, the curve. No.
No.
Maybe the slides are not moving on your end, so. Yeah, because we are only seeing the first slide.
Yeah.
You're only seeing the first slide.
Yeah.
Yes.
Now you don't see anything else.
No. If you can close that panel, maybe.
Okay. All right. You don't see anything now?
Just the same, just the same.
Close the slide completely and come.
It's okay, we see now.
You see now?
Yes.
Yes.
Is it moving now? If I move to the next slide.
Yes, it's moving now.
Okay.
It's moving.
Thank you. It's just two slides that shows the interaction with the environment and how complex. And I think you all appreciate the complexity of cancer, which is the ultimate complexity, biological complexity you can ever have. Now you have three main I call it catchphrases, or it may be paradigms that is now quite important, which is the biological networks that we have seen in the past presentation, the feedback loops, and then personalized precision perspective. And I will try to go over that quickly in the time allotted to me. We started a long time ago, if some of you were not born yet, looking at the genetics of cancer. That was a few years after the discovery of the BRC1 and BRC2. To our surprise, there are two different studies done in Sudan by two different groups. One is us and the other is Italian, that the breast cancer, the BRCA1 and BRCA2 mutations are not that common in India, and it was verified later. However, there was a study around that time, and it seems that there is a lot of mutation, that truncating mutation and others that are found in high frequency in Africans, yet it did not translate. And that's 1999, about four years after the discovery of the BRCA1 and BRCA2. So that the conclusion is BRCA1, BRCA2 mutation really does not mean a lot. And I will show how later on, why. Then we turned our attention to the mitochondria, because we were working on the mitochondria at that time. And we were looking at mitochondria as a surrogate of genetic diversity. And the figure on the right is, it shows you the amount of genetic diversity that you encounter in East Africa, in particular Sudan, Ethiopia, and Eritrea, which is almost 60% of the entire genetic diversity that you can have globally, that is witnessed globally. And this has been also verified now by other genomics study and a recent publication by the Oxford group. We looked at peroxyredoxin because it was found in a proteomic study, and Also, it showed that there are differences between what you see in Africa and what you see in China, indicating again that this variation that people have mentioned in the previous studies. But and then we thought, what if it is something that has nothing to do with, if it is a mutation are caused by the environment. And this came through one of the clinicians who in our group who noticed that some families with nasopharyngeal carcinoma have also breast cancer. So he suspected EBV. And the conclusion is, it's a long story that it was published about 10 years ago, that Epstein-Barr virus is a major culprit of breast cancer in the Sudan. And the positivity rate is around 100% in the tumor, but not in the safe margin. So that was confirmed by in situ hybridization. And we looked at epigenetics, as something that can explain the absence of somatic mutations. And this is a very old also table, which indicates that when the genomics analysis were not yet that common, we used simple methylation techniques. And again, breast cancer had a completely different profile from colon cancer, nasopharyngeal carcinoma, esophageal cancer, sorry. So we went, and then the age of genomics came into being, and we did a whole methylome analysis on breast cancer cohort, and it showed that indeed the reasons for the BRCA1 and other genes, BRCA2, loss of function is epigenetic silencing, and that was very common, as well as other genes that you can incriminate in the. So there were a story now, and a story that relates Epstein-Barr virus, and I will come to that later on also again. But when the age of genomics, when there was dawning of that age, we started doing exome sequencing on individual patients, on group of patients, and it came immediately clear that the mutational profile in Africa is completely different than what you can encounter. And it's directly related to the effective population size, the extreme diversity that we find in Africa. So the idea of having a minor reference allele with a certain frequency that is become pathogenic, that might not apply if you have an excess of minor reference allele. And that was a major guidance in our analysis of subsequent analysis of genomic analysis, whether exomes or whole genomes. We applied that immediately to a family of an extended family of colorectal cancer, and that was published about 8 years ago. And the interesting findings about this family, first is that the conventional tumor suppressors or other cancers are not the main what you call driving force. We used one of the earliest analyses, I think, that has used a system-based approach, a network-based approach. And the genes that came out were mostly RNA binding proteins. And these RNA binding proteins are now emerging as a major also target of studies and analysis of cancer. And I will show why. One of them is ELAVA1 or HUR, H-U-R, and this is a very important gene. But the more interesting is that when you look at this family, you find more identity by descent, less identity by descent, identity by state. That is to say, there are two brothers that are being analyzed in this family, but the genome of these two brothers looks less identical than the genome of one distant relative to another cancer patient. So the two cancer patients shared more genetic material than the brothers. And this is very important. And the reviewers refused our interpretation. It was published later under a different title, which is that it's an organizational issue. So cancer is an organizational mechanism. So cancer is organized. And now, there's a paper now that came this month, Nature Genetics, about how important is networks, that central networks. It's not only one gene, but genes that share networks probably are the target, are supposed to be the target of our intervention. So now comes the the issue of personalized medicine. If you look at this family, so the profile of those two brothers is different, is common, but different from other members of the family. Yet also the prognosis and the sequelae of the cancer was also different between the two brothers. And all is shown in the genomics in the genome of those two brothers. So why is RNA binding proteins are important, and why are proteins that alter the genome, not only tumor suppressors, are more important? If you look here at these networks, these are the same family, and you look at the controls, you see how complex is the networks of the cancer patients compared to the networks of the controls, of the genes of the controls. And this is due to a very important protein, which is the histidine deaminase, which does what HUR also does, which is it incurs mutational changes across genes. And that's where you see the complexity of cancer. If you look at the hallmarks of cancer and their variation, this is due to this kind of intervention. Add to that microRNA. So the question of regulation is so important. And so this paper talks about regulation. And that, for example, one of the genes is regulated by like 30 genes and microRNA. But it is also a feedback mechanism. So this gene also regulates the regulators. And as I mentioned earlier, one of the major, now you call it dictums of modern biology, is this feedback loops. I'm about to finish. So this is just so this microRNA that has been established in this family. This family has been extensively dissected for everything. One of them is microRNA 29. So microRNA 29 is a homologue of Epstein-Barr virus. So now we have something that we can summarize here, that Epstein-Barr virus, Epstein-Barr virus is quite common globally. It's a very common virus, but in Africa in particular, it is associated with several cancers, not only breast cancer that we have shown, nasopharyngeal carcinoma, probably colorectal cancer. And the idea is Epstein-Barr virus, through either itself or through homologues, affect genes like ApoBec or histidine deaminase that carries out all these differences.
I'm Montasio. Can we make some conclusion quickly? Because
conclusion is Epstein-Barr virus is a target for vaccination, fortunately, and this is where the translation comes. We are in contact with a group in Germany who have developed a vaccine for Epstein-Barr virus for infectious mononucleosis. And if it proved possible, if it shows any promise, then we can have a prophylactic vaccine against not only breast cancer, but other cancers. Thank you very much.
Thank you, Montasir. I'm sorry to cut you short, but the time we need to give the other participant time to ask. So please, if you have questions, Francis will take over from now and for questions and answer, and you can raise your hand. We have 10 minutes, or you can put your question on the chat. And Francis, is yours, the floor.
Thank you very much. Thank you very much, Salma. I think we have our, from the charts, we have a question around technologies that are used in sequencing. So maybe we can comment on that. Pamela was concerned about the questions, had a concern on the technologies. So I think Solomon has tried to answer that, but maybe for the benefit of everyone, I'll ask Solomon to respond to that. And then also at the same time, I'm asking Evelyn to respond to a comment and question from Elizabeth, who congratulates you for the presentation and agrees that African tumors should be studied and by Africans in Africa and stored in Africa. So she's like, what plans do we have to store our samples right here in Africa, and as we study and analyze everything in Africa? So I think maybe we'll talk about capacity building for us to be able to do that, and also we have the capacity to do that, or what plans do we have so that we can, all our studies are in Africa, we don't have to ship the samples outside. So I don't know who wants to go first. Solomon, you want to talk about technology, and then
yes.
Evelyn can talk about the, please
yes.
go ahead.
Yeah, thank you very much, Francis. As you rightly noted, Pamela and I, we've responded, but Pamela wanted to know if we're able to, if you can use Oxford nanopore technology or Sanger sequencing for RNA-seq. And so if you look at the whole RNA-seq, when we look at the sequencing all the over 20,000, maybe 30,000, genes that are expressed. And so you need a panel-based set technology to do that, where you have the genome, printed on on a chip, and you can read that. And it's Illumina that has that kind of technology. And there's another company, coming out of China now, which is MGI, also have that kind of robust technology. Again, there are other technologies that can do RNA-seq beyond that, but not, so definitely not sequencing. And Oxford Nanopore is not robust enough, in my own opinion, for that kind of study.
Okay, so thank you. I'm taking the question on keeping African samples in Africa for research in Africa. And that is something that I have been trying to, different methods of preserving tissue for, I guess, the past 5 to 7 years. It depends on what you want to do with, ultimately, with the tumors that you are trying to store. Keeping tissues in buffered formalin for 6 to 48 hours and then making a block out of it is the best way if you want to sequence the tumor later, both for RNA sequencing and whole exome sequencing. But the challenge is making sure you have access to pathology so that the tissue doesn't stay in formalin for more than 48 hours. And that is a challenge because many institutions don't have readily access to pathology services where the tissue can easily be converted to a block. And once the tissue stays in buffered formalin, or some people don't even have buffered formalin. So once it stays in formalin for more than 48 hours, then the quality of your sequencing goes down. Another method for preserving tissue is using the tissue fix and the tissue stabilizer. So with that, you can keep the tissue in the stabilizer after keeping it in the tissue fix for 24 to 48 hours. That one can stay in the tissue stabilizer for up to a month. That is if you're able to get any regular fridge and put the tissue in there. So that is a very good resource for Africa where you don't have ready access to pathology. But then when you have a cold chain system where you can keep the tissue in -80 in liquid nitrogen, then of course that's the ultimate if you are hoping to do sequencing. And that is very expensive and not everyone has access to that. But when we are writing grants and when we are collaborating with other institutions, we are hoping that they will make these facilities available to us. But before then, the best way again is to make sure that these tissues that if you need them for sequencing, then try and keep them in formalin not for more than 48 hours and then make a block out of it, or tissue fix and tissue stabilizer. If you want to do like work where you are going to need fresh tissue, then you have to have the tissue slow frozen in -80 and then transferring it to liquid nitrogen. And that one too is you have to have the facilities to do that. Not every institution has it. And so again, if you have a central location, maybe within the country that can be done, that will also be helpful. And these are some of the SOPs for doing this. We are going to have it on the basic and translational research page where we'll have the method for tissue processing and tissue storage that you can easily have access to that on our page. And so that there are some of the processes that are relatively inexpensive. And I have my pathology colleagues online. We have been trying different methods to get this done. But again, formalin, tissue fix, and tissue stabilizer is your best resource in Africa for now. Thank you.
Thank you, Evelyn. I think there's another question to you from Faith Zaplov. And she says you mentioned about cell lines specific for the African population. So she's wondering if there are any cell lines, such cell lines available in the market, or do we have any lab that have Africa-specific, Africa population-specific cell lines that one can access?
Yes, so in a recent publication, we outlined the PDXs and all the cell lines, everything that we have developed mainly at that time from the Ghanaian tumors. And we are now developing it. Now we have Tanzania samples that we are going to begin to develop them, and they are available for collaboration. And so you can email me after this, depending on what you are doing, and we can make available some of these resources. But again, for now, these are we have for Ghanaian samples. If you are interested in making cell lines or PDXs, I don't know where you are from your tumors, we can also help you with the procedure and what it will take for you to do it, so that you'll be able to do it in your institution. And when it is done, you have to validate it. You have to prove that what you have done is really representative of the original tumor from which it was developed. So you always have to make sure you keep some of the original tumor so that it can be validated, and then you can use it for research and it's publishable. And so there are procedures for doing that, and we'll be happy to share that with you as well.
Can I say something before we continue, Francis? I see people starting to leave. Please leave your information if you want to be part of this group. You can send it to Bill, your information, or you can put them on the chat and we will contact you. Go ahead, Francis.
Thank you. I think our time is up, and I don't see any questions apart from Nasur Bunyinza, who is just thanking the speakers for the good job. So I'll want to take this opportunity to, on behalf of the organizing team, to say thank you very much for all of you, to our presenters who are the founders of this group. I want to say thank you very much for joining, for the presentations that you have done, all the people who spared their time to attend. I say thank you very much. We want to invite all of you to join this interest group so that we can take, this collaboration. We can catalyze ourselves to study the cancers in our population and also try to find out solutions to our populations as much as we can. So want to thank you very much for attending this. Let's keep the conversation going. I'm sure the organizers, the members of the interest group will come up with another webinar and they'll update us or inform us on when that will be. So thank you very much and have a blessed time. Thank you.
Thank you, Francis.
Thank you.